So the third part of the lecture, I'm going to talk about drug discovery based on the virus dependence on the host cells, as well as our immune responses, which I call the resetting part of this relationship between the virus and the host. As I mentioned, a antiviral therapy can either target the virus itself, such as targeting the viral enzyme, which this so-called direct antiviral therapy, this virus direct antiviral drugs that targeting virus target, they are specific to certain viruses. So that's good. So they are like, say they have a good specificity, but associated with that is the problem of drug resistance to occur, because when the virus can easily mutate its own genes to evolve drug resistance. In addition, for newly emerging viruses, viral epidemic, such as COVID-19, when a new virus comes in, it takes a long time for you to develop a specific antiviral drugs. So like in the case of COVID-19, you might, when you really have a good drug, you might no longer have patients to treat. Another strategy is we can target the host targets that are required by the virus infection. As I just mentioned, there are many host factors in human cells that are required by the viruses. Those factors, they might be shared by many different viruses. So by targeting those common target, we might produce really broad spectrum antivirus, meaning a drug can treat many different kinds of viruses. And also, since the antiviral therapy target human target, it's very difficult for the virus to evolve drug resistance, because the drug doesn't bind to a viral protein in this case. But there's some concern that if you have drug targeting host target, it might affect the normal function of those targets and cause potential harmful effect. So these two methods, they are pro and cons in each category, and they actually are quite they are complementary to each other. Give you examples of targeting the virus. Since the therapy targeting viral target, they are quite common. I'm here, since I'm talking about virus host interaction, I'm giving you more example on the host targeting strategies, which can really benefit from the study of virus host interaction. So as you probably all heard, the drug chloroquine or hydroxychloroquine, these are old drugs developed for treating malaria, but during the study of many different viruses, people found that the chloroquine really have a broad spectrum antiviral drug, at least in cells and in animals. In the case of COVID-19, unfortunately, the clinical trials didn't show its significant positive effect. Nevertheless, chloroquine has been demonstrated to have a broad spectrum antiviral effect in cell culture. How does it do this? Chloroquine, as I actually block this viral entry process called endocytosis I mentioned to you. As I just mentioned, after the viral particle is uptaken by the endocytosis process, there's a downstream step which involves the acidification, the decrease of the pH of the endosome, so that it becomes materialize into the so-called later anosome. This low pH is critical for the fusion of the viral membrane with the anosome membrane. Only after the membrane are fused can the viral particle be released into the cells. And the chloroquine can block this acidification process. And that is actually at least part of the mechanism of its antiviral response to block the entry of of many different kinds of viruses, providing a broad spectrum antiviral effect. Another strategy of targeting the host cell is through targeting the host cell metabolism. This is an experimental drug, and it has shown named S416. This inhibitor was developed as an inhibitor of an enzyme called dehydroorotate dehydrogenase, which is a key enzyme in the human RNA synthesis. As you know, RNA are assembled from the building block called a nucleotide building blocks of RNA synthesis. It require this building block also require a synthetic pathways, and the enzyme DHODH is involved in this synthetic pathway. And this group of studies show that these DHODH inhibitor can have a very potent inhibitory effect on COVID-19 replication in cells. While it has it certainly also can affect cell growth at high concentration, but the viruses is much more sensitive to this inhibition, providing certain specificity of this using this drug as antivirals. Besides targeting host factor, the receiving part of the virus-host interaction, we can also targeting the resisting part, the antiviral immune systems. Since it has found that in COVID-19 patients, a very big problem of this infected patient is the so-called cytokine storm. Basically, the virus infection lead to a massive release of cytokines by the non-cells or immune cells at the non, which really cause a very serious side effect. So in 2 studies actually show that if you can use antibody targeting one of the cytokine called IL-6 to block this cytokine storm, basically this is basically an over response to this overreaction basically of our antiviral response, which really cause a harmful effect. So we can actually control this inflammatory reaction by using antibody targeting interleukin-6. As I mentioned earlier, cytokines are proteins that are released in the extracellular environment. So if you inject antibody, you can actually block this the cytokine storm and ameliorate the patient's reaction to viral infection. This is an example of treating COVID-19 patients using antibodies that modulate our immune responses.